Welcome to our discussion on Neisseriae. These bacteria are quite distinctive as they appear as paired round or oval cells known as diplococci. They have a unique reddish-pink color after Gram stain when viewed under the microscope, which helps us identify them easily. Understanding their structure is the first step in appreciating how they behave and cause disease. Let's start with the basics. Neisseria bacteria are gram-negative cocci, meaning they stain pink and are shaped like small spheres arranged in pairs with flattened adjoining sides. They're non-motile, so they don't move on their own. Two main species to focus on are Neisseria gonorrhoeae and Neisseria meningitidis, both important in human infections. To understand Neisseria better, we look at their general traits. They don't have flagella or spores, but do have capsules and pili, which help them attach to host cells. They need oxygen to survive and produce enzymes like catalase and oxidase. They are pathogenic, meaning they cause disease and prefer environments rich in carbon dioxide. They can't survive long outside their human hosts, which makes them strict parasites. A key point about Neisseria is that they infect humans only. Both men and women can carry the bacteria without symptoms, which makes tracking infection tricky. That's why contact tracing is crucial to stop spread. The bacteria attach firmly to epithelial cells, especially in gonorrhea, where they multiply and cause infection. Now, looking at the two main types, gonococci and meningococci share several features. Both are pathogenic diplococci and produce endotoxins rather than exotoxins. Meningococci are good at colonizing the back of the throat, while other Neisseria species usually live harmlessly on mucous membranes or skin. This similarity helps us understand their relatedness and differences in disease patterns. Despite their similarities, gonococci and meningococci cause very different diseases. Gonococci cause gonorrhea, which usually results in localized inflammation, mainly in the urethra, and is rarely fatal. Meningococci, on the other hand, cause meningitis and other systemic diseases that can be life-threatening. This difference is critical for diagnosis and treatment approaches. Let's explore what makes Neisseria gonorrhoeae so effective. It has several virulence factors. Its surface antigens can change to evade the immune system, a loosely attached capsule that prevents phagocytosis, and pili that help it stick to host cells. It also has outer membrane proteins important for infection, endotoxins that cause fever, and an IgA1 protease enzyme that helps it establish infection by breaking down immune defenses. Gonococcal infections can lead to serious health problems, starting with gonorrhea, which presents as urethral discharge. It can progress to pelvic inflammatory disease in women, causing scarring of fallopian tubes and risk of ectopic pregnancy. Inflammation can also affect ovaries and fallopian tubes or prostate and testicles in men. Newborns can suffer eye infections leading to corneal scarring, showing how this infection affects multiple systems. Turning to Neisseria meningitidis, its virulence factors include a capsule that protects it, pili for adhesion, endotoxins that cause toxic effects, and an IgA1 protease that breaks down immune defenses. Classification into 13 serotypes is based on capsular polysaccharides, including types A, B, C, Y, and W135. These features work together to help it invade and cause serious disease. Meningococcal infection starts with colonization in the nasopharynx. From there, it can invade the bloodstream, causing meningococcemia. It spreads directly to the central nervous system via olfactory nerves or the blood-brain barrier, multiplying uncontrollably in cerebrospinal fluid. This can lead to Waterhouse-Friderichsen syndrome, a severe adrenal failure, and other infections like pneumonia and arthritis. Understanding this progression is vital for recognizing and treating meningitis. Laboratory diagnosis relies on collecting samples like pus or swabs from the urethra or cervix, and blood or cerebrospinal fluid, depending on the infection site. Simple and gram staining techniques are used to identify Neisseriae bacteria, which appear as gram-negative diplococci. Accurate diagnosis is essential for effective treatment. Growing and identifying Neisseriae requires specific conditions. Selective media and enriched blood or serum provide nutrients. They thrive in capnophilic environments, meaning high carbon dioxide levels. Modified Thayer-Martin media is often used for selective growth. Tests like oxidase and serological assays help confirm the species, allowing precise diagnosis and guiding treatment. Managing Neisseria infections involves antibiotics like azithromycin, beta-lactams, tetracycline, and quinolones, each effective against different strains or species. Vaccines also play a key role, especially against meningococcal infections, providing immunity and preventing outbreaks. Combining these strategies helps control and reduce the impact of these bacteria. Finally, non-gonococcal urethritis, or NGU, is caused by a variety of agents beyond Neisseria gonorrhoeae. Bacterial causes include chlamydia, trachomatis, and others, while viral agents like herpes and cytomegalovirus also contribute. Fungal infections like Candida albicans and protozoal infections such as Trichomonas vaginalis are involved too. Knowing these helps tailor diagnosis and treatment beyond gonorrhea.